All right, so back from coffee, so people have a higher level of awareness now, and we need that because we have Bechir Jarraya here, and Bechir is an MD PhD. He's a neurosurgeon, so if anyone has any problems, talk to him, and a professor of therapeutics at the Medical School of University Paris-Saclay, and he's also a principal investigator at Neurospin, a neuroimaging center. But we know Bechir very well also from different projects we have been developing. And what is really fantastic about Bechir is that he sort of succeeds in bridging, let's say, this deep understanding of the neurological side of consciousness and states of consciousness, but in parallel developed really advanced and sophisticated methods to investigate the principles, the neural principles of anesthesia at the system level. And this is really unique because most people look at these kinds of phenomena either from a very large distance in a correlational approach, or you look at it from a very microscopic perspective, or in isolation from a clinical perspective. And Bechir really brings all these perspectives together in a comprehensive view that he really translates into working experimental paradigms in the macaque monkey. So it's really a unique contribution to the field of consciousness studies that Bechir is realizing. And I'm really looking forward to your talk, Bechir. Welcome.
Thank you. Thank you very much. I would like really to thank you, Paul, for this invitation. I love this format of International Summer School of Neuroscience. I want to thank Anna and Ruth for the organization, and thank you to be here numerous during holidays to have a summer school. So good to be here. My talk is actually kind of consequence of the previous one, and it's entitled "Can Neuromodulation Restore Consciousness?" You can see this artistic painting, and you can guess a macaque monkey thinking, maybe being aware of what's happening outside, when having these nice pyramidal neurons all around. So this is thanks to Marietta Wren. She's an artist in Paris, and she made that as a logo for our team. I want to thank her again. I profit from that, from the questions we had previously. There's a lot of work we described this morning that is done in monkeys and non-human primates. And of course, this is an ethically challenging aspect because these are animals, and animal research can raise some ethical issues. Of course, everything is in accordance to ethical committees and whatever, that's the minimum. But we are very careful about using the right model at the right time, meaning if you can address a question without going to animal research, of course you should privilege that. And I agree with you, if you can do some experiments in humans, again in the ethical framework of clinical research, then obviously you should address the question directly in humans, in volunteers or patients. But sometimes some questions will need a specific model. And I think as a student, I encourage you to always ask during your experiments why I am using this model, why I'm using rat, I'm using monkey, I'm using human, I'm using just a computer as a model, or whatsoever, and is it relevant or not? Now, in this talk, actually, there will be three parts. First, what is consciousness? Although now you had already several talks about, but I will focus on what we call disorders of consciousness. Then, what is neuromodulation, and specifically something we call deep brain stimulation, but not only. And could we combine these neuromodulation technologies to hope to reverse consciousness disorder and restore consciousness to some patients who miss the consciousness after severe trauma, for example? So what is consciousness? You could probably notice from yesterday and today that it's a polysemic word or ambiguous word because you can use it for different meanings. And I want to make just some clarifications. They are to distinguish what we call state of consciousness and the content of consciousness. State of consciousness, for example, this sentence, she remained conscious after the car accident. So She is not in coma after the car crash. She's still conscious. So this is the state of consciousness. And we refer generally in that to wakefulness, to arousal, to vigilance. And this, you can observe the transformation, and when you go from sleep to wakefulness to anesthesia to coma, this is something very, very, very remarkable. Conscious access is more subtle. For example, I would say she was not conscious of the red light. That's why she had a car accident. So it was in her visual field, but still it was not accessing the conscious content. And here it's rather about awareness. It's quite different from the first case where we're talking about vigilant person, wake person. And it's about a specific processing of a piece of information and its broadcasting across the brain. In terms of neural models, we have the exact same differences in terms of neuroanatomical models. So for vigilance, for state of consciousness, mainly the main systems are actually deep-seated systems, started from the upper part of the brainstem or mid part of the brainstem, called ascending reticular activating system, that projects to the thalamus, midbrain thalamus, while the conscious access will rely on a main cortical network, and this refers to the theory of global neural workspace previously described, which is made of frontal, parietal, and I would add singular cortices processing. See, for example, if I see a stimulus, like the red light that was not seen by the lady driving, the stimulus was still on the visual areas and had some feed-forward propagation, but it was, for some reason, was not strong enough, was not pertinent enough to have a general reverberation, broadcasting aspect, meaning that this piece of information will also interact with attention networks, with memory networks, with all the subjective part of the person. And of course, both systems are related. So the ascending reticular system and the thalamus are pushing and incoming activating information that go widespread through cortical areas and are key to guarantee this broadcasting effect. Is that clear enough, or are there questions at this level? Yes? So please, you can ask questions during the talk. You can see it here. For example, I will put on, maybe you saw this graph yesterday, no? Yes, you are familiar now, from the Liège group. Aurore Thibault was here yesterday, and you see that vigilance here versus awareness, you can distinguish both aspects that are completely different, and you have a set of disorders. So coma is here, 0, 0. General anesthesia, we don't know exactly. It's something that you could see much less simple than we thought previously. Sleep with the different sleep levels. Something important to distinguish, and we talk mainly about these states, what we call vegetative state and MCS, minimal conscious state. You see that vegetative state patients wake up after a severe injury or severe stroke. They open eyes. Family think that they are already conscious, but unfortunately it's not. But they have absolutely no interaction with the external world, whereas MCS, minimal conscious state people, will be between the normal awareness and the zero awareness, which is vegetative state, and very importantly, they fluctuate. MCS patients fluctuate a lot. This is almost a characteristic of these patients, to have fluctuation. Vegetative patients have no fluctuations. You could see also that modern imaging, such as fMRI, can be a way to enter in contact with these patients who have generally paralysis, and it's very hard to know if this person is vegetative or MCS patients. Even you can try to interact with this person. The lack of response doesn't mean that this person is vegetative. By doing so, we have 40% of mistakes. And an elegant way, described by both the group of Cambridge and Liège a few years ago, is to have this paradigm of tennis imagery versus spatial navigation imagery. And you could see that this patient, who was stated as vegetative state patients, could actually respond and have some kind of reportability. And I agree with Paul that with the local global work, we had no reportability in the monkey. But actually, you see here, it's a kind of fMRI reportability. And we accept that in this category of patients as a way to report on what is going on outside the world. Now, what is neuromodulation? This is again a polysemic word, an ambiguous one. I can say that because if you look at literature, there's extending literature on neuromodulation, and there are really two different meanings. The very first meaning historically was this one, is about describing some neurotransmitters like dopamine, like catecholamine, who inside the brain do not transmit information from neuron to another, but rather will make an ambience, an atmosphere of increased level of this dopamine neuromodulator, for example, that will change the excitability of other synapses without transmitting directly information to these neurons. But you can see also this other meaning of neuromodulation, which is more at the system level, which is more requiring devices or pharmacological agents, like stimulating the brain, stimulating the spinal cord, or administering genes through viral vectors to influence whole networks of the brain. So for the first meaning of neuromodulation, we're talking about small circuit, and the neuromodulator here is tuning the excitability of the synapse, and the synaptic property, it's acting like a hormone, basically. It's much slower than the rapid synaptic transmission, of course, and have a diffuse form of excitability enhancement or depression in whole area or regions of the brain. Now, the second meaning, which is more a system-level neuromodulation, you will see all these devices, electrical stimulation, like deep brain stimulation, TDCS, I think you saw yesterday again, TMS, magnetic stimulation. So all these are now more and more routinely used or in the clinic or in the experimental setup in the lab. Optical stimulation with optogenetics, which is a rising technique in neuroscience, mainly in animals so far. Ultrasound, but also pharmacological neuromodulation, and gene viral vector modulation. If you look at in terms of, I just made a PubMed trial and put the word neuromodulation from the 70s to now. And you see that there's a surge in papers talking about neuromodulation, and actually it's rather in the second meaning of this term. Okay? And we'll see that there's some convergence between the two ways of considering this neuromodulation. Let me show you some examples because this is a summer school, including example outside of the consciousness field. So for example, pharmacological neuromodulation. If you take cocaine, now we have a strong understanding about where cocaine acts on, how it increases the amount of dopamine in the synaptic level in the striatum, for example, how it affects dendrite structure in the medium spiny striatum. But another way to look at the cocaine effect is actually on a brain-wise, on the global level, and not only on the striatal level, because the striatal action have a widespread diffuse action. And again, this is challenging to do in humans, although many volunteers will pop up, probably. But ethically challenging, it's not because you can do it in human that ethical committee will allow you to do so. And it's really remarkable now to be possible to train macaque monkeys, and they generally love to do that, actually, because they go outside their cage, they go to the scanner, they have stimuli, they have juice coming here to reward them. They are inside a chair, but they can move their body freely. They are not really completely constrained. But by doing so, you can see, this is the work I did with John Arsenault a few years ago. You can see both strong inhibition and strong activated areas just by the administration of cocaine, by contrasting fMRI data on the cocaine during cocaine administration minus the resting level. And you see that there's even a dose effect in terms of general circuitry. These are really very precious information because you cannot obtain them, of course, from naive healthy volunteers. You can also do more complex experiments and have conditioning between cocaine and a visual stimulus and show that just the visual stimulus in monkey without cocaine administration can strongly activate orbitofrontal cortex. And again, this whole level neuromodulation can give you some ideas to potential targets to intervene or cocaine abuse. Another example of neuromodulation, which is gene-mediated neuromodulation, another disease, Parkinson's disease. It's a disease where you lose your dopamine here in the brainstem, neurons producing dopamine, so this pathway would disappear. Dopamine is a strong neuromodulator in the striatum, so this will disappear, and this neuromodulation will disappear, and then you can replace the loss of this neuromodulation by means of viral vectors that are injected in the same parts that are missing dopamine, and they can strongly affect neural activity within the circuitry and also brain activity within basal ganglia once the viral vector is working. Of course, this translates in behavioral improvement in Parkinsonian models. Now we'll move to more common neuromodulation means, which is a direct. Yeah, sorry.
So the gene modulation, is that already established? It's not used yet in humans, right?
It is. Now there is a clinical trial going both in Paris and the UK. Yes. And there are already first preliminary results that are really encouraging in terms of safety, and there is some efficacy already, yes. But these are very long, there are new generation of this viral vector, so it's a very long clinical trials, actually, yes. So let's go now to electrical stimulation and something called deep brain stimulation. I don't know, is everybody familiar with DBS or deep? No? Okay. Yes, there are some experts there. All right. So let me show you a little bit an example how you see here in the brain of these patients 2 electrodes, 2 leads with different metallic contacts that will be wired and connected to a kind of pacemaker that delivers current. And the delivery of current can be made really very locally. For example, here in structure called subthalamic nucleus, deep in the brain, because this nucleus will have a burstic abnormal activity during Parkinson's disease. So I show you just an example. This is a Parkinsonian patient, and you can see him shaking strongly, and as soon as you put DBS on, it was previously implanted, of course, you see an immediate correction of movements. This person is a university professor, and he volunteered to give his videos for other patients. So you see that neuromodulation of a very tiny part of the brain can have a strong effect on the general motor control. So this was described first by Professor Alim Louis Benabid in Grenoble. Other people helped developing other targets, of course, in Oxford, Boisous, Malon Delon, and targets now move to what we call subthalamic nucleus. This, the device is made of electrodes like this that are inserted inside the brain during a multi-hour surgical operation, because the accuracy of lead placement is key, of course, as you can guess. This is related to a pacemaker that goes under the skin, so everything is completely hidden. And this can be programmed by the outside. Patient can still have some check through a remote control. Importantly, what actually Alim Louis Benabid described clearly is that you need to have a frequency of stimulation more than 100 Hertz to have DBS working. This is a surgical procedure. So patients is it's an invasive neuromodulation, of course. Although there is no lesioning of the brain, this is almost a whole day surgery. And patients have this kind of frame to maintain the head completely fixed. MRI now is making strong progress to show the targets. This is the Parkinson's disease patients, subthalamic nucleus. You can record from these structures before inserting leads. You can even test during the operation if patient is wakened at that time, and insert final electrodes with completely inner device. Good, but how can we improve this technology? And actually there's a lot of research now. First, to use more and more ultra-high fields, and at least 3 Tesla. And this week, for the first time, Siemens have CE approval for 7 Tesla as a clinical magnet. So for the first time now in the future, we will see in hospitals 70 MRIs for routine clinical work and not only research as so far. More and more there's research about having this stimulation not only working constantly, but reading the brain, reading the implanted area or another part of the brain to modulate, to change the rhythm, the intensity, the programming of the stimulation according to the brain activity of the target or of cortical regions that are key for the mechanism of DBS. The leads are also more and more sophisticated. Look at, so far the leads were sending current as a sphere, so they were making a whole modulation that sometimes go beyond the nucleus, and sometimes you have incredible psychological effects during DBS. There were description of depression, that on-off depression, which, by the way, made new findings about brain networks involved in depression. But still, today, you can have the possibility to have current steering with segmented leads and send the current, and more and more to have a computational model, actually, to have a more control on the shape of current diffusion. So this is really very, very, getting very, very powerful in the future. And that's, I think, very important, including for our talk, the restoration of consciousness. Waveforms also are improving, and so far you can have a constant stimulation with uniphasic, with biphasic, or alternate uni and multiphasic, and actually this the waveform can matter, can change the behavioral effect. Other developments, could we do DBS without inserting electrode? This is the team of Ed Boyden at MIT, who released very recently this very nice paper in Cell, showing that by stimulating superficially with a small difference, small delta, so temporal interfering electric fields called technique, they could, with superficial electrodes, perform a deep brain stimulation here in the hippocampus of this mouse, for example. And you see that only this hippocampus was stimulated without affecting the other cortical areas on their way, on their pathway of the electric interference. So technology is really making strong steps and can make these DBS much more smarter in the future. Of course, there's all the optogenetic field. In that case, the optical DBS would be made through a laser, and you need, of course, to have your neurons previously sensitized to your laser. And the way to do that today is to deliver genes like channel rhodopsins, halorhodopsins, RHT, there's a lot of them actually, with viral vectors that make your neurons or your pathway, your cortico-subcortical pathway, sensitive or extremely sensitive to very specific wavelength of laser, and they can activate or inhibit your target. This is so far, optogenetic DBS is made only in animal models. Now, what is the mechanism, actually? There's a long debate about how can we transform a completely shaky Parkinsonian patients into somebody like you and me with the fine-tuning of motor control by just sticking electrodes deep in the brain of basal ganglia. There was like almost 20 years of debates on how DBS works, and there was a kind of empirical saying that DBS were at high frequency were inhibiting the target, which was controversial because how do you explain that stimulation becomes inhibitory? Actually, recent work now is making progress into the mechanism understanding, and more we are making the discovery that the answer is not deep, but it's superficial. Look at this paper, the group of Philip Stark at UCSF, published in Nature Neuroscience. In these patients, you see these are actually patients who have DBS electrodes. These are Parkinsonian patients. And during the surgery, they introduce, through the bar hole, there's a small craniotomy here to introduce the electrode. They also introduce this cortical grid, small cortical grid, to record cortical activity. And they did sessions where they stimulate deep in the brain, the subthalamic nucleus, while recording in motor areas that we now project actually to this structure. And this is the very interesting finding. Actually, here you have spectrograms that will show the phase amplitude coupling, or you probably know about the oscillation theory with the cross-frequency correlations and phase amplitude coupling. Is everybody familiar with that? Okay. Actually, what you need to imagine is that in these structures, you have we can identify oscillations, and what you could see here is that, especially on the beta, which is around 18-20 here hertz, we have a strong correlation coupling between the phase and the amplitude, meaning that the beta oscillation will entrain the amplitude of firing rate of these cortical areas. It means that cortical areas is locked by the pathological oscillation of subthalamic nucleus. And what happens, so this is a measure comes from the data recorded in the cortex and not at all deep in the brain. And you see, so this strong phase amplitude coupling. When you do DBS, so really 7 centimeters far from here, you reduce dramatically phase amplitude coupling. You make, again, firing rate more independent from the phase of oscillation of subthalamic nucleus, of neural activity, sorry. So, and this is actually starts to be back when you stop DBS. So it looks like what happens when you do DBS is a strong cortical configuration, reconfiguration, especially in that case. This is not fMRI here, this is actually electrophysiology, so we cannot have assertion about what's going on across the brain, but this have been addressed recently by the work published yesterday, I think, with Gustavo Deco and the London team in patients with fMRI. Second paper showing that DBS is actually about cortical modulation. It's this one, the group of Carl Desrot in Stanford. In this paper, actually, they modeled Parkinson's in rats. They did subthalamic nucleus stimulation electrically. Well, it works, it corrects Parkinson's. But if they do optogenetic stimulation of this nucleus, it doesn't correct behavior at all. But if you label with viral vectors the cortico-subthalamic, the cortico-subcortical pathway, and you stimulate optically this pathway, you have the correction of Parkinson's. It means that DBS works mainly by the reconfiguration of cortical areas, by retrograde modulation from the subcortical area to the connected cortical area. This is really very elegant demonstration, and the nice thing with optogenetic is with electrical DBS you stimulate all the neurons and the fibers in the area that is stimulated. With optogenetic you can label with genes only the targeted area or the pathway that projects to this area or goes from this area. And you can nicely dissect the stimulation of each compound and demonstrate that it's actually the cortical part that is key to explain the DBS effect. So actually, it's deep stimulation, but it's a superficial effect. It's large-scale cortical effects. Now this is really revolutionizing the way we understand how DBS works. And this actually, so if this is really true, if we perform imaging, like fMRI, while we do electrical DBS, then we should see strong cortical modulation. Okay? You agree with that? You would predict that. So far, it was extremely tough to do in patients, Parkinson's patients, because the devices were not MR safe. Now it's changing just recently. So we did this experiment in monkey, and you could see that you have a DBS here, and you have a strong bilateral cortical activation with fMRI when you do, for example, a basal ganglia stimulation, going in line to what we previously described. So the combination of DBS or microstimulation and fMRI can be a new way to describe really what's going on in the cortex. And we know that most of the mechanism of DBS is about cortical modulation. You can even see the difference between, we did that previously, with Hiro Iomuro, that the frequency effect, there was the debate, why should we have high frequency more than 100 hertz to have DBS working? You see that if we do fMRI in this monkey at 20 hertz stimulation or 120 hertz, you see that only higher frequency have cortical effects, while low frequency have mainly local basal ganglia and even cerebellum effects in terms of fMRI activations. Okay, so at this point, we can say, just to conclude on the neuromodulation part, two definitions, but by the end, both neurotransmitter neuromodulation or device neurostimulation end up affecting large-scale networks beyond the first target. DBS affects massive cortical, sometimes even bilateral networks to achieve its behavioral effects. And the combination with fMRI could be of a great value to distinguish the proper effect and to develop maybe a new DBS and eventually for consciousness, which gives us to our talk. So I show you again this figure. Now you are really familiar. And what happens in these patients, mainly MCS patients? You recognize him probably. This is a famous MCS patient, so Michael Schumacher. I will not talk about non-invasive neuromodulation because yesterday you have a very nice talk about TDCS. So I really will talk about DBS. So because we want actually to restore global neural workspace in these patients, we need a technique, a way that can have large-scale cortical modulation. And DBS looks to be a good candidate, as we have seen already in other diseases. So the hypothesis is, are there some brain nuclei somewhere in the brain that are good candidates for DBS to restore this frontoparietal singular reverberation of information in the brain of MCS patient, for example. So actually the topic is not that new. This is 1969. Hassler in Verona found this paper when doing really extensive literature on that, and I was surprised that there were even photographs. You see these patients with tracheostomy, so probably in an intensive care unit after weeks of trauma. They call it apalic syndrome because at that time it was really very hard to distinguish vegetative, minimal conscious state. It was, everything was almost put together. And they already put an acute electrode. It's not, there was no DBS devices for chronic stimulation, but they put an electrode already in the thalamus, ventral anterior thalamus part. And they show that under DBS, patients have some improvement in behavior. And they did even some EEG recording without the fancy methods we have today to analyze signal. But still, it was really very, very interesting observation showing improvement, transient improvement, because, of course, they could not keep the system. There were no DBS devices at that time. Years ago, Coadon and Richer in Bordeaux did use at that time the DBS device, because at that point, historically, there were development of DBS devices to treat pain, central pain, okay? Before, much more before Parkinson, DBS for Parkinson's. And they had this idea, and they published this paper, deep cerebral stimulation patient with post-traumatic vegetative state, okay? I personally don't believe that we can wake up a true vegetative person with DBS. But at that time, again, when they stated vegetative state, very probably some of these patients had minimal consciousness state. As you know, even today, there are still errors in labeling VS or MCS. And they showed that this time they put electrodes in other part of it's still the thalamus, but other parts in centromedian parafascicular complex. Why CMPF? So CMPF is what we call intralaminar nuclei inside the thalamus. And these actually, this complex, CMPF, have strong and massive, widespread frontoparietal singular projections from the thalamus. And they showed some recovery of some degree of consciousness in almost half of the cases. It's very hard to guess who will benefit and not. And almost at the same time, the Tokyo team, Yamamoto, in 1991 and then later on, continued doing actually DBS, thalamic DBS. Again, same target, CMPF. So 70 cases in, if we add all these publications. And for the first time, they started doing some imaging. You could see here that after stimulation, this is hemodynamic, CBF, in the brain, or glucose consumption of the brain that increases with thalamic DBS. So they already could show that thalamic DBS can enhance strongly widespread cortical network activity. But the results were really very variable. And no way, there is no way to have a real conclusion from that today. Again, part of the problem is the distinction, the real distinction between VS and MCS. And then we come to this paper that becomes highly cited, but you should remember that previous work also was done. The New York team, the Schiff paper, this is one patient with MCS 6 years after head trauma, and he was still having a low level, very low level of awareness. So this is a CRSR scale. And you see that he was implanted in what they call centromedian thalamus, which includes also centromedian parafascicular complex. It's very hard to distinguish all these nuclei because you don't see distinction with MRI. And in these patients with highly deformed brain, because of their head trauma, it's very hard to use atlases to co-register this brain with atlas. The nice thing with this paper is that the first time they did a control, internal control, which is patients was observed on the on state and on the off state. And they could show an increase in arousal, an increase in feeding behavior. So multiple items of the CRSR improved transiently and in a reversible manner with thalamic DBS, showing for the first time more specificity. Now I can show you, so the same team of the previous paper performed an experimental work with to try to precise more which part of the thalamus is important. And using this time optogenetic in rat model, they could show that in anesthetized rat, they used the anesthesia model to model loss of consciousness. They could show that optogenetic DBS had an effect, again, by direct thalamic stimulation, mainly on high frequency 100 Hertz. It's really amazing. We find the same high frequency requirement with in optostimulation as compared to electrical DBS. And they did, for the first time, fMRI in rodents, showing that they have strong manipulation with 100 Hertz optogenetic in the thalamus, a strong manipulation of cortical large-scale networks. Doesn't mean that they restore consciousness, of course. It's very hard here to know. But it's a demonstration of large-scale manipulation by thalamic stimulation. So what was very tempting is to move on with to refine these targets in the non-human primate model, in the macaque model, using the anesthesia model that was thoroughly described in the previous talk. And I can show you, for example, this data. So exactly the same setup that Lynn showed previously, the same levels of anesthesia, resting state and task fMRI. These are just to remind you the kind of dynamical connectivity that we could study, and we did with stimulation. Lynn showed you that by the end, when we contrast the data we have in our monkeys between the awake state and the anesthesia state, and if you take the global effect contrast across all these data, we find that there is a strong activity that is specific to the awake state, or that is much stronger, including the intraparietal sulcus. It's actually the fundus of IPS. It's called VIP. And they are prefrontal cortical areas. And deep on, you have this activation that corresponds exactly to parafascicular central median. So, by taking this, of course, you can very quickly say this could be, for example, potential targets for electrical stimulation. And again, we chose to go rather to the deep area because although DBS is about, now we know it, about manipulating cortical areas, it's much more powerful to influence cortex through a deep structure than covering the whole brain with electrodes, almost, to be able to reproduce the same effect. So this is a previous work from my postdoc, Naoki Tani, from Japan, showing techniques to implant and to use high-field anatomical data to implant DBS in monkeys. So we implanted this monkey that you could see. This is the artifact of the electrode. It's much bigger than the electrode by itself. It's MRI safe, but it's still generating an artifact. And you can see that you can also, and that's very important, to you can model exactly the contacts of the electrodes within an atlas. So this is thanks to suit imaging called lead DBS, lead neuroimaging. It's Andreas Horn in Boston and Charité de Berlin who made it. And we did a co-development for a monkey box, a macaque toolbox within this solution. You can see here that you can really see each contact within the thalamic area and also simulate and model the volume of activated tissue. Because according to your parameter of stimulation, you can have only CM, CMPF, CMPF plus CL. Thalamus is such a highly fragmented and parcellate nucleus that any DBS there can solicitate many nuclei at the same time. And I can show you some initial data. So we are using exactly the model that Lynn described, with the monkey having controlled anesthesia. It's deep anesthesia with EEG monitoring, because the dose of anesthetic is important, but to achieve the same deep anesthesia, it's never the same across animals. It's like humans also, right? You don't need, probably if you go for surgery, the same amount of propofol to achieve the same level of depth of anesthesia. And I can show you a video, for example. This is outside the MRI. And you could see that if you program from the outside the stimulation in this monkey, who was profoundly anesthetized, very suddenly he starts waking up, having strong movements, even the tail, but the hands. And if you go in the front, actually you see also facial expressions. And if you take it back to off, everything goes immediately. So you have an on-off effect. It's there is no seizure there because we have EEG, so it's not a seizure induction. It's a really strong arousal. You can see it also that monkeys stop using the machine to breathe. It's exactly like end of the surgery where patients are going out of anesthesia. So, and this was really highly reproducible. So the question was, how can we understand what's going on there when we do DBS in the thalamus to induce vigilance and arousal in this monkey model? It's not very trivial, actually, but we could do this kind of experiments, and it's part of the project of Jordi Tassri, PhD. And you could see that you have the monkey inside the MRI here. You have this 8-channel coil around, and you have special cables that allows the electrodes to cross the room and goes to the control room to have an external stimulator, because the internal devices cannot be completely driven when you are inside the MRI. Okay, so with the animal we have the advantage to have external stimulator and the possibility of having exact synchrony between the fMRI acquisition and the stimulation. And you can do, for example, a block design stimulation, so resting state, having 30-second stimulation on, 30-second stimulation off, different parameters of stimulation, different contacts of the electrodes. And, of course, you can do exactly the same with the local global paradigm on top of it. And you can see here that when you have the wakefulness effect of DBS, and only when you have this effect, you have actually a strong frontoparietal activation in the macaque brain that you don't have when you have a non-wakening paradigm of stimulation. So definitely DBS of the thalamus is about a strong, large-scale cortical restoration. Second, if you look at it in terms of matrices, you can show that you induce again some anti-correlations that disappeared under anesthesia, and also some strong correlation that disappeared, but also anti-correlation, which is really very interesting, even though we still don't know exactly the meaning of these anti-correlations. But again, this is a matrix of cortical scale. So the steps that are ongoing with these experiments is that we are continuing the dynamic resting state fMRI acquisition with different stimulation contacts and different stimulation levels, because the best control is also to have a DBS stimulation that does not induce the wakefulness effect and not only the complete off. Second is, do we induce just vigilance or do we induce awareness, actually? And here it's very difficult, of course, in this animal model to do, but again we will use, and we are using, exactly the same local global paradigm that you have seen this morning. And I can show you very early data with Lynn on local global paradigm played to the monkey when the monkey is on stimulation, on DBS, so anesthesia, propofol, on DBS versus off DBS or DBS that is at lower rate, that is not making any behavioral effect. And you could see that we start to have prefrontal and parietal activation specific to the DBS that induces the behavioral effect, meaning that probably we are inducing a restoration of, between brackets, global neural workspace of the monkey, although he have strong propofol sedation. And essentially, and very key, is the parietal part, we talked about it this morning, that we're missing under propofol, and that is being restored with DBS.
And this is unilateral stimulation?
Yes.
Right.
This is unilateral stimulation. It's true that in macaques, even more than in humans, we have very quickly bilateral effects. For example, we see it with globus pallidus or basal ganglia stimulation. We see the two motor areas that are activated with unilateral. Last but not least, going more to modeling, to computational model with Gustavo here, with Morten here. And here the idea is starting from this model now of loss of consciousness with the local global paradigm, with the DBS within the thalamus, we may build a theoretical framework of induction of consciousness under anesthesia in the macaque model. And in that case, we could predict with the model maybe other targets in the brain that could work even probably much better or that are easier to target practically. And that would be really a fantastic step in the future because it would both reconfort the model and being a new way, a new paradigm, even in the neurosurgery field, to generate targets for DBS, for diseases, not based on historical cases, but completely on a theoretical model, which probably would be a revolution in the field. Okay, so very probably we are again doing what we have seen in Parkinson's, meaning DBS in the thalamus, but large-scale frontoparietal singular effects through this DBS. And I will show you just a video now what happens in humans. This is a particular situation. This patient have tremor, and he went to surgery for tremor, and it happened that the target for tremor, not Parkinsonian tremor, it's called essential tremor, is within the thalamus. It's not centromedian parafascicular exactly, but it's close by. It's ventro-intermediate VIM nucleus. But in the end of this surgery, if you look at this patient, before waking this patient, he's still under strong sedation with propofol again. We put the stimulation on with a volume of activated tissue that goes a little bit beyond VIM to CMPF. And you see that we are inducing exactly like the monkey reaction, awakening reaction, while he's still having strong anesthesia levels of propofol. And actually, anesthesiologist was completely frightening. Oh, what should I do? Should I increase anesthesia? No. Just shut down the stimulation. Again, you make the effect disappearing right immediately. This is what Lynn at some point mentioned, what we call bispectral index, BIS, which is a measurement, rough measurements of EEG, frontal EEG. So to conclude, I would say that anesthesia is a powerful mean to provide a model of altered consciousness in non-human primates and to study not only the mechanism of general anesthesia, but to develop neuromodulation and electrical neuromodulation, especially with DBS in our case. And that thalamic DBS probably could restore not only vigilance, but something higher level than vigilance, because we are seeing the restoration of the parietal component of frontoparietal singular GNW. And thus, that DBS holds really potential to restore consciousness in these patients, these MCS patients. But I think that we need to improve the targets and to use improved technology as compared to the previous papers, including the steering technology to be more specific within the thalamus. And I want to thank very much all my colleagues and collaborators, because as you could guess, this is work that requires multidisciplinary interaction. And to recall that CETA approved all these protocols, of course, in monkeys. Thank you very much.
Thank you, Bechir, for this very, very interesting talk. So we have now some time for questions. Of course, we have one for Paul, for sure.
So, Bechir, so thank you. This is really fantastic. But now if we look at the macaque result that you showed, right? So on the one hand, behaviorally, we have some twitching, let's say, right? So this is one question. As a critic, I could say, well, there's some reactivation of, let's say, midbrain brainstem behavioral structures, but it's not really targeted towards the thalamocortical system, so we don't see reactivation of consciousness or anything close to it. And then you might say, well, but look at the correlation structure of the activated regions, right, in the fMRI. But if you really compare it in a bit more detail, the kind of structure that emerges is sort of closer to the propofol pattern than to the wakefulness pattern, right? So there are two issues. So why do you believe that the behavioral signatures you see are indicative of, let's say, level of consciousness changes? and on what grounds do you believe that the correlation structure you see after DBS is closer to the awake state than to the propofol state?
Thank you, Paul. It helped us a lot to write the paper because reviewers will ask for that for sure. So first, for the behavioral effect, something really interesting that happens beyond these movements that again are not seizures, because we had EEG and we questioned that, was it just seizures? And it's not. We have really documentation of EEG of all that. Is that not only there are these motor effects that really looks like these are human by the end, but also they have all what anesthesiologists see when patients is emerging from anesthesia. So they breathe differently. They stop relying on the machine to breathe, whereas under propofol they could not. They have an increased heart rate. They behave exactly. I mean, I think Lynn, you could witness about that. It looks like we are waking up completely somebody. It looks like the recovery period of anesthesia. So of course the fMRI is not enough to say that it's that might be that some awareness there. As a strong difference between the propofol and the propofol plus DBS is the parietal reintegration. This is again, I think, is really key, because we never saw reaction to the global effect in the parietal under propofol with all the data we collected in several monkeys, while here it is reappearing for the global effect. One of the ideas we have in the future also to do the same experiment under ketamine, because with ketamine there was no global response at all. But there is clearly a difference with the propofol, even though it doesn't look so on the prefrontal part.
But the signatures you're describing, like breathing, heart rate, and so on, do not necessarily depend on the thalamocortical system. They would depend more on midbrain brainstem structures.
Sure.
Right?
Sure. That's why it wasn't enough at all. Actually, one of the big challenge we had is what do we make of that? What does it mean? And the only way we could find to move on, I'm not saying we have the final word, is our local global paradigm again.
Changing because the EEG, when you look at the EEG, because we are calling the EEG, during the stimulation you have really deep anesthesia signal of the EEG, and when you stimulate the monkey at the parameters for waking, when you have the clinical awakening of the monkeys, then you go to an EEG signal which is described for like a very light sedation. So,
okay.
Yes.
All right.
On how many patients did you try this experiments with the propofol?
5.
5.
Yes.
They were all Parkinsonian patients?
No, 3 will have just tremor, called essential tremor. It's a tremor without any Parkinsonian symptoms, and 2 are Parkinsonians. Because
I know that deep brain stimulation with certain patients, Parkinsonian patients, doesn't really work. For instance, so this intracranial electrode stimulation is not successful in all patients.
For tremor, you mean? Sorry?
With Parkinsonian patients.
Yes. In the thalamic, in the thalamus, you mean?
Yeah.
Yes, actually, in very, very few cases, there are Parkinsonians who have 90% of their symptoms are really tremor, and almost very, very few rigidity. And in very, very few, there are very few cases where we did DBS in the thalamus for Parkinson's, like 25 years ago. But you are absolutely true. This is generally it's subthalamic nucleus and not thalamus.
Okay. Some more question, anybody? Then we pass to the next speaker, and we thank again Bechir.